with nucleosome as the basic unit, you can see here, each of these nucleosome on the histones, they also have little tails that's kind of dangling outside. And these tails can be modified. And depending on the different modification, if you imagine strings on, beads on a string or pearl necklace, you can imagine some of the pearls have a little flag. And because the eukaryote genome, especially you can imagine the human genome is huge. And for a transcription factor to be expressed and going to the nucleus, it's really quite blinding. It has to really quickly find where to bind out of 3 billion locations. And imagine having to go through a lot of locations and trying to see where this transcription factor motif exists so that the transcription factor can bind there. It takes a lot of trial and error. So histone modification or epigenetic in general, you can imagine are beacons in the genome which tell transcription factors or other factors where to go first. And so very interestingly, this histone modification is a very, very powerful beacon in the genome that tell the transcription factors where to go. And as we mentioned in the nucleosome, there's like 2 rounds of DNA and there are 8 different histones. And there are 2 histones called H2A here and 2 H2B and 2 histones H3 and 2 histones H4. And they are kind of 4 together and they stack on top of each other. Actually, I don't think this figure is accurate. It should be the 4 with 1 circle and another 4 with the circle, not the other this way. Anyway, and you can see here, this is the histone tails that comes out of this, and these can be modified. And based on the different modification, you can also specifically enrich for locations in the genome that has the histone modification. And this can be done with ChIP-seq. So you can imagine if we fragment the chromatin, remember in ChIP-seq, we fragment the chromatin into, say, a couple hundred base pair fragments. And normally for transcription factor binding, you just use an antibody against the transcription factor. But once the chromatin is fragmented, we know if the histone tail is modified, there are antibodies against modified histones. You can use antibody to kind of collect all the pearls that have the little flag and then isolate the DNA that's wrapped around that nucleosome and sequence those out. Then you will know which location in the genome the histones are modified. And so this is called histone mark ChIP-seq. And so in the genome, these little marks can be added by readers or readers. They can also be removed by erasers and also be read by histone mark readers. And so depending on the different location, and there are also different marks. And this is just one example. There are modifications also on H2, also H4, but this is just one example of the histone H3. Position 1 is on the is at the end in here, and as you go inside, this will be more like a position 79. And so you can see here, most of the modification happen on the arginine and lysine. R is arginine, K is lysine, or threonine T, and also serine on P, and also maybe on P locations. And so depending on the different locations, for example, you would very often see something called H3 lysine 4 trimethylation. It means that on this tail at this 4th position, because there is a lysine, you can see there are many different modifications that can happen. And in this position, if there are 3 methyl marks, that's called a H3K4 trimethylation. If there is only one, so basically on the carbon of this lysine, if there is one methyl mark, that's monomethylation. because the carbon, one end is attached to the remaining amino acid, and then the carbon has 3 hydrogens. If one hydrogen is replaced with a methyl, that's monomethylation, Me1, 2-methylation will be methyl 2, and trimethylation will be Me3. So all 3, hydrogen bond are being replaced by methyl groups. So that's H3K4 trimethylation. Another one that's quite popular is actually not really indicated in here, but on this lysine 27 position, it can be methylated, but also can be acetylated, and that would be called an H3K4 acetylation. And so this will help you know how to read different histone marks. And in here, you can see there are many, many different names in here. These are the readers or writers or erasers. You can see if there is a stop sign in here, that means it's a lysine demethylase. Whereas if it's black, it means it's a writer in here. And also in this case, it's a H3 lysine 36 writer. Is called lysine methyltransferase at this location in here, which will add, you can see it's a very complicated mechanism. There are many, many writers, many, many erasers, and many, many readers, which add and remove these histone marks. And there's also something called a chromatin remodeler. So remodeler, place the nucleosome into different locations. They kind of help position them or help slide the nucleosome along the DNA. And writers add the mark, erasers remove the marks. Readers can recognize the marks, and usually by the reader, after the recruitment or after the recognition, it will try to recruit other factors such as transcription factors, and also help recruit additional writers and erasers. And so epigenetic in the cell is in a dynamic process, right? So these kind of writing and removal and reading and recruitment really happens all the time. And when you do a ChIP-seq, you just capture a snapshot of what's happening in millions of cells. And so there are some clarifications. When we talk about DNA, it's really the naked DNA double helix without any higher or without any structure. But when we talk about chromatin, it means that it's DNA in its compaction structure in the cell, including nucleosome, including the higher order compaction, and also the other modifications. Nucleosome is the basic unit of the chromatin. It's 146 base pair DNA wrapped 2 rounds around 8 histone proteins. And for regular transcription factor ChIP-seq, you use sonication to fragment the DNA. You use the antibody against the transcription factor that binds to the DNA. For histone mark ChIP-seq, you can use sonication, or you can use micrococcal nucleus. Basically, you cut on all the nucleosome-free regions, and you collect all the nucleosome like pearls with the DNA still around it. And the antibody is against the modification on the histones. And if you look at also the nucleosome sequencing here, you sequence all the nucleosomes, and you can see here only occasionally the nucleosomes are well positioned, but in many other locations it's kind of randomly spaced. Whereas when you look at histone modifications, in this case, if it's micrococcal nucleus treated, you don't really sequence all the nucleosomes. You only look at nucleosomes that are modified, having specific modification. And so this is kind of a cheaper experiment. Instead of collecting all the pearls, say you only collect the red pearls or pearls with a red flag. And in here, you can see the signal here is much, much stronger. You still see the same positioning of the nucleosome. And you can see here, this one has a very strong, like a very strong flag. And the adjacent one is kind of weaker, because usually when the histone mark writers go there in the right position, it will add the mark. And then, like nearby things get like modified a little bit, but they're not the main players. So the strong modifications actually indicate strong signals, and that they send signals to, say, when a transcription factor is available in the nucleus, it will look for locations where these marks are, and it can find the locations much, much faster. And so these are much higher resolution. It has better signal to noise. The difficulty is there are many different marks. And so every histone mark ChIP-seq is a separate experiment.